welcome everybody and do a quick roll call, and that way we'll know who's been able to join us. So, and then Nicole will jump in as well. So everyone, this is Andrea Epstein, and I'm here with a number of members of our Global Genes team, including Nicole, Kim Kilborn, our VP of Advocacy, Carrie Austria, our Director of Advocacy, and Carol Dutch, who is a volunteer but leads one of our corporate alliance committees, as well as Katie Mastro, who is managing all the logistics associated with our call. We're pleased that we're all here today. We know it's been a while since our last call back in the fall, and we apologize for the delay in getting this group together. Many of you have participated in some of our ad hoc discussions between November and today. We'll be just doing a brief recap on this as well. So the first part of the call is the Global Genes update. Then we'll go into just some recaps on the ad hoc initiatives. And then similar to last time where we had the Global Alliance for Genomics and Health do a member presentation with Peter Goodhand. Today we're delighted to have Ethan Perlstein of Perlstein Lab to give that member presentation and share some of the insights and the science going on with his labs and initiatives. I'd like to just do a quick roll call and ask. It looks like we have 15 attendees, but some of them are hard to tell. So we know, Ethan, you're on the line. Christina, Ron Taft, are you on? We have you on.
Andrea, it's Aisha. It's the best voice I've gotten through, so I'm listening in.
Okay. Others, we'd like you just to jump in because you're coming up. We have a number of participants, but we don't have names. So Harry, we know you're on. We know the Global Genes team. Others on advisory board, so you could just jump in and let us know that you're on.
Raft.
Hi, Hud.
Hi.
Good morning.
Hi.
All
Janet
right.
McCarthy. It's Janet McCarthy here. I just have to say that there was another number that was sent out a while ago that called into, and a couple other people were on that number wondering if it was the right one. So I'm not sure if we can resend the recent email out with this number.
Okay.
This is Global Genes. Kate Brown was on that call with Jeanette and I, and she didn't get a copy of the email, so I forwarded it to her, but I don't see her on the invite list.
This is Aisha. I chatted with Kate yesterday, but I think it's the WebEx that led to the new number, so I'll send another info out right now.
Okay. And Katie also sent an update, so hopefully Kate will be able to join us shortly. And Jeanette, thank you for letting us know. I think with with our time sensitivity here, we are going to jump into the Global Genes update, and from there, hopefully, if others join on, they'll be able to kind of jump into the ad hoc initiative. So, with that, we're going to move to the next slide. Next slide.
Okay. So hi everyone, it's Nicole. I know we just have a couple of brief things to share with you today. But one was Rare Disease Day, World Rare Disease Day, that was celebrated February 28th. There was a lot of activity around the globe. We distribute ribbons to different organizations, different hospitals, different affiliates, different nonprofit organizations, patient groups, et cetera, for different types of events, and you can see the statistics there. It's grown every year, and, we believe that, this collective effort in trying to raise awareness and make a difference is starting to gain traction, and we also participate in a week-long event in D.C. over 200 advocates, and we kind of stormed the hill to promote World Rare Disease Day. And then in addition, we'll be working on expanding efforts where World Rare Disease Day on the last day of the month in February becomes a kickoff to efforts in March. And our ultimate goal down the road is to continue to build partnerships with consumer brands, et cetera, so that we can start raising more money and awareness for rare disease around the globe. So, again, successful, and we have some other activities that happened that one of our guest speakers today will share as well.
So, great, thank you. Glad you're able to join. We've just really very brief introductions, and we've talked a little bit about World Rare Disease Day, but we're just getting into the meat of our presentation, first half on Global Genes Initiatives, and the second half on a presentation by Ethan Perlstein. So I'd just like to introduce RAREMed Forums and introduce Carol Dutch, heads our healthcare practitioner committee from our corporate alliance. RAREMed Forums is kind of it's a newly branded program that evolved from our initiatives focused on grand rounds that began ago, but really kind of have blossomed during this World Rare Disease Day. So without kind of going into too much detail, I'd like to introduce Carol.
Thank you. Thank you. Thank you, Andrea, and everyone for giving us this opportunity to share with you this very next initiative. The subcommittee that I co-chair, our objective is to work on creating events, firms, tools to the healthcare professional community to help them with raising the awareness about rare diseases and where we can fill in gaps that other avenues haven't been able to do with rare disease information diagnosis. So the RAREMed Forum has really, as Andrea said, been launched this year. And what we've done is we've created platforms where we can have our patient advocates able to share information about their particular pathology, about their organization that will help the medical community perhaps manage their patients that they have already diagnosed, but also help with patients who have not been diagnosed, sharing symptomology and experience that they've had. This initiative is some. I would just say that this initiative started because we did do a survey amongst the healthcare professional committee or community to really find out that there is a lack of knowledge with a lot of the general protection community to know how to handle a rare disease patient, hence created these forums. Do you want the next slide? So the quarter of this year, we've already had 3 RAREMed forums, and we tried to have them around the World Rare Disease Day to kind of celebrate that event. However, we're not saying that they have to occur in that time period, and you'll see that we're planning to do more. The first was at Children's Hospital in Orange County at Chalk. That top picture shows the audience, and Andrea Epstein also spoke at the event. We had about 100 to 120 physicians, social workers, genetic counselors present. This particular case, it was part of Chalk's Pediatric Grand Round program, so there was CME credit that was given in this particular event. The second forum that we did was held at Rady's Children. I don't know if Keith Fox is on the line, but he was the sponsor of that, and you can see his photo right there in the bottom picture. There, we had the opportunity to have the UCSD residents and medical students at that event. 2 patient advocates who are part of the Global Genes membership speak to the audience. One of them is with the Association of Creatine Disorders, and actually had her 2 children diagnosed at UCSD, as well as Julie Cunningham's child who has immunodeficiency disease, and Dr. Hal Hoffman, who is her physician, was at the event. The third forum we had was at Children's Hospital of King's Daughters in Norfolk, Virginia, so on the other side of the states, and that had 40 to 50 genetics and medical residents. So we were, continuing our platform in having the patient advocate community together with the medical community, again, in sharing information about diagnosis and raising awareness about rare diseases. So plans are to continue this. Those are the smiling faces of 2 medical residents. We're very happy to receive the toolkits that we distributed at the UCSD event. I can share with you that one of the comments that came out of the crowd, which was very nice to the patient advocate, was one of the residents saying, I'd be asking, you if you're in the ER. And the patient advocate said, ask me anything and everything. We just want to be able to share our information, and that, you're wanting to learn more about our specific epidemiology. So in closing, I just want to say that we're trying to work on getting sponsors and getting these programs up for Toledo Hospital, CHOP Philadelphia, Nemours Children's in the Delaware site, and Cleveland Clinic. And in order to have this program go launch out on a wider basis, we're really writing kind of a toolkit preparation. Katie Master has been excellent in setting up packages, checklist, an introductory letter. In other words, anyone who's interested in sponsoring this, we're able to kind of give you our knowledge, the information, so that it's easy to put together a RAREMed forum. So if anyone has any questions, or especially if anyone is interested in sponsoring and putting on one of the RAREMed forums at your particular institution, we'd love to hear from you. Thank you.
Thank you. Thank you so much, Carol. And definitely, if anybody in our group is interested in spearheading a Grand Rounds type of program focused on rare disease, we would be happy to provide you with the resources that we have deployed for these programs, and we can touch base on that after this call. So thanks again, Carol, for a great overview. Moving on, our big initiative, and I will let Nicole jump in on this as well, but this is our, just briefly, we just wanted to share with you, kind of make sure it's on your calendar to be participating and joining us for our next Patient Advocacy Summit on September 24th and 25th back in Huntington Beach, the same place that many of you attended last year, both for either part of the summit as well as the tribute event, as well as our initial kickoff for the Medical Scientific Advisory Board last year. And a couple things to point out are just that this is a 2 full-day meeting this year, where there's lots of new kind of content being added to the program, which I'll just let Nicole jump in on, but we do have a strong science focus this year, and there are going to be more opportunities for deep dive discussions, as well as really getting more kind of honed in on what the patient and the advocate's role is in drug development.
Yeah, just so a 1-minute overview. Last year, HUD participated, and some other patient advocates, et cetera. Our last session was around drug development. Kind of the key participants involved, and we'd gotten a lot of feedback from the attended that said we want that woven throughout the conference. But we have a heavy We have heavy content and different tracks related to drug development where patients play. We have different tracks on details and specifics related to, things you need to know once you have a diagnosis. What do you do? What are the 10 next steps you need to take if you're interested in participating in funding research? We have stuff around consent, data. We're going to include additional science deep dives. And one thing that we will be delivering there, we have 3 toolkits around early clinical drug development and 2 others related to drug development, including a visual map that is being created as well with the focus on where kind of patients play and the role that they play in the drug development sector or in drug development. And so we will send you the working agenda, and we'd love to get your feedback. And as always, if there's areas or opportunities where you think you, know someone who might play a key role as a potential participant on the agenda, and or if you yourself think that, that you would like to participate, let us know. We have many of the spots. We have invites out to participants, but we are currently continuing to work to fill it. So stay tuned for more, and we're excited to see you guys in September.
Great, thank you. All right, next I'd like to introduce Kim Kilborn, our VP of Patient Advocacy, who's going to share some information about the role that Global Genes is playing in the 2nd annual David R. Cox Prize for Rare Compassion, which is actually a motivational incentive program to catalyze medical students to kind of focus their career in the area of rare disease. And we will be also inviting members of the Medical and Scientific Advisory Board to participate as on the review committee for those winners of this prize. So with that, we'll introduce Kim to share a little bit more about what this exciting prize is.
So thank you, Andrea. We're very pleased to partner again with STAND, which is the Student Advocates for Neglected Diseases. This is a program that was started by Colton Margus, who is a 2nd-year medical student at New York Medical College. He has a very personal connection with rare disease. Two of his brothers living with AT, and that sort of inspired him as a medical student to form this group, also to create this particular prize. Recognizes or really sort of encourages medical students to connect with a rare disease patient and really learn more about them and how that will sort of inform them as they grow in their studies in the medical profession and then ultimately practice in the field. And that relationship that they will build with a rare patient, and Global Genes helps connect them with rare disease patients as well, is to write about that experience and in the form of an essay. It's a 2,500-word essay. This year we are fortunate to be able to offer 3 prizes for the essay contest, 1st, 2nd, and 3rd place. And medical students will be asked to make those connections with their patients and write about that over the summer and submit their essays through our website on Global Genes sometime, I think it's August 10th, to be considered. Now, the prize was named in honor of Dr. Cox. He was a professor of genetics and pediatrics at University of California, San Francisco, and then later on in his career at Stanford. And he was really committed to and showed tremendous compassion, even outside of having a personal connection, but was very dedicated to the area of rare diseases and especially in the field of AT. Slide, please. So again, students are asked to forge a relationship with a patient or family and write about that experience, and that scope would also include strategies for improving physician-patient relationships and building that partnership. and broadening their understanding of the unique complexities of treating rare diseases, and hopefully spurring their interest in pursuing a career in research and development and treatment of the many rare diseases that have been identified. And next slide. So the submission guidelines are on our website, globalgenes.org/Cox Prize. Building about this over the summer. And in terms of the submissions, they will be evaluated. There's certain criteria that we've put forward, and we're working with Colton and his group STAN to raise awareness and sort of get the word out within the medical community. We have a number of promotion and delivery objectives that we're following to make sure students are aware of this. We also this year have broadened it also to the UK and Canadian medical students to participate. Evaluation will be a first round by members of the Global Genes leadership team. And what we will do then is sort of call down the essay entries into a number of finalists where we think the essays are particularly strong. And then we ask that a few members of Medical and Scientific Advisory Board participate in helping us further narrow down and select the first 3 prize winners in this essay contest. We'll also engage with some of our patient advocacy leaders in the initial evaluation. The prizes will be announced during the Patient Advocacy Summit and during the Community Awards presentation. Hope that that some of you might consider participating with us. We think this is a great initiative to help, raise awareness for rare diseases, especially among young medical students and physicians that are entering these fields. So thank you.
Great, wonderful. Thank you, Kim. And we would just ask that you spend a little time thinking about whether or not you'd like to be on the evaluation team for these awards. We're probably going to seek about 4 to 5 members of the Scientific Advisory Board to participate on the evaluation and selection committee, and a separate outreach, asking for volunteers will be coming out shortly after our call today. So you can look for that as well. With that,
this is Steve Croft. Can I just make a comment?
Sure.
Okay. I participated in this last year, and it was really an enjoyable experience to read the descriptions by the students. So, I would encourage others to join in this year. And the second point is, I did cut Colton up with this student group in Europe, Student Medical Association in Europe, and in the future, perhaps with the association with Urordis, that Nicole has been able to work very nicely. Maybe this is something we could extend over to them to also take a similar approach with the rest of Europe, for the students who might want to participate as well. So reaching out might be very helpful as we move forward. So that would be it.
That's a good idea, Steve. Thank you.
Hi, this is Kate Rao. This is Rowan calling from UC Davis. With regards to that award, I'd be very happy to serve on. I knew Dave very, very well.
Okay, wonderful.
Wonderful.
Thank you so much.
We'll send something out, and we definitely appreciate that, and we'll definitely include you on the going forward. And thank you for your comments. So we're going to jump into a very brief update on the initiatives of the ad hoc groups. I know, next slide, let me just see. I think we're going to do this a little bit out of order. We're going to go to the patients ad hoc first, because Harry Leiter is going to be kind of providing some insights, and I know he's on a time crunch, and then we'll move back to the other 2 initiatives as well.
So Harry, go ahead. This is Nicole. So Harry's going to kind of set the tone as to, kind of how this project came to be, kind of the outcome of our discussion during the committee call, about a month ago, and then Andrea and I will give an update as to the actions that we took post the committee call and kind of where we're at with this initiative. So please, thank you, Harry. Go ahead.
Sure, thank you much, Nicole, and I apologize I can't stay for the whole meeting. Some of the updates and some of the initiatives that you guys just went over are just wonderful. I particularly like the student contest. Yes. In terms of this idea, the idea started with a notion that all rare diseases in aggregate are common, and I think Nicole and Andrea and the whole Global Genes organization has done a phenomenal job of awareness around that fact. An individual, a parent often, who has a child with a rare disease, it doesn't look that way. So we know from some of the work that's been done in one study, people have or their parents oftentimes see many providers, many doctors, have many tests, and eventually they reach a rare diagnosis, and it can often be months or years before they actually get a diagnosis. Obviously, sometimes you never get a diagnosis. The thinking was, how do we accelerate? Could we create a tool that would accelerate the recognition that you or your child might have a rare disease? The idea would be a screening tool, like for those of you that are familiar with mental health, something like the PHQ-9, which is a 9-question questionnaire, which is used throughout the world to screen for depression. And if you answer yes to a certain number of questions, you have a very high likelihood of having depression before you even see a clinician. And if we could create this kind of tool and make sure it's valid, a questionnaire that might predict you have a high likelihood of having a rare disease, maybe because you've been bounced from doctor to doctor, or you've been tried on multiple therapies, or no one's been able to give you a clear diagnosis, whatever it is. It'd be potentially put on a link to something like Walgreens' mobile app, where the millions of consumers who our app every day could literally pull up the tool, fill out the questionnaire, and if they were potentially, if the tool indicated might have a rare disease or their child, it could link them to resources like the Global Genes website or the hub we're talking about or other resources. So the idea here is there's an unmet need in the market to provide the population with a quick tool that might alert them that their child has a rare disease. The idea is simple. Levels in the details, and what we need to do if we executed this was to actually create a survey and validate it. And I think that's where Nicole and Andrea have done some further exploration on how we might do that. Is that correct?
Yes. So thank you so much for that overview. So yes, you are correct. So we went out to, so we ran this by a couple other folks. I know Dr. Groff, we had a little discussion around this as well. So we brought this idea to a professional that does these types of marketing surveys, and kind of shared the idea, shared kind of what the goal and the outcomes would be, and she had suggested, we need to see if there's any FDA regulation around these types of questions, if it was ever created into a handheld tool or, made available widely. And so the answer was no, so we're good there. And then we met with, actually at World Orphan Drug Congress last week, with someone who's on the data aggregation and analytics side. And so between this woman, and I think some of you may know her, Patty Engel from Engage Health, who does this for biotech and pharma. It's her career in terms of conducting these market research polls, and then working with David Lapidus from Lapidus Data. We had kind of broached the idea. And did everyone on the team get the overview that we put together by chance of the kind of like the executive brief of this? Yes. Okay. So you should, in your inbox, kind of the executive brief that we put together related to this program. On the committee, okay, so the rest of you we can send to if needed. So we want to move this forward. We want to do it right. I know that the committee had said, even though that there's some market research that has been done, Shire's Rare Disease Impact Report, Global Genes actually did another research survey with Patty Engel and her team as well for a corporate alliance. So there's data out there that we can use as a starting point. But in order to make this something that can be widely used and respected, we just want to make sure that we cross all of our I's cross all our T's and dot our I's. Because, ultimately, like Harry said, it would be nice to have this be available in, Genome Magazine or Good Housekeeping, where people, it's not to take away, so just to clarify, it's not to diagnose patients. It's to really get people saying, I think something's going on, and where would my next best resource be, right? Andrea will share with you an undiagnosed portal that we're building out. We have a ton of things in the works so that we can build this hub that becomes a viable resource for individuals that are looking to be connected. You may be, last thing I'll say is you may have a rare disease, but you may be, like many people, kind of stuck in the disease silo, kind of feeling like you're still isolated and alone, even though you do have a rare disease. And a tool like this could also get them connected to the larger, broader community and be able to benefit from resources, that Global Genes and other organizations are creating as well. So it's kind of a next step, and we will have some more information and thoughts and insights from both Patty Engel and David Lapidus on kind of next steps as we pursue, doing this market research poll. So,
great. All right. Well, we are we have a jam-packed schedule here, so I'm going to jump into a brief update on the programs group, and then we'll be able to give Ethan Perlstein the time that we committed so that he can share the innovations in his lab. But just real briefly, any of you who participated in our kind of first, almost like focus group discussion last September about kind of what things the Medical and Scientific Advisory Board could work on, one challenge that was presented was what we should be doing with our undiagnosed pilot program, where we have in the last 2 years raised funds to sequence through exome sequencing primarily 30 patients who were on a medical odyssey to find a diagnosis. And the net at the end of kind of this pilot, we still have a couple stragglers that have not been able to get their sequencing done. But we, on the one hand, we found that we were really only helping probably 25 to 30% of those families get a diagnosis. The rest of them ended up with a variant of unknown significance. At the same point, on the other side, we found that our donors who had helped seed fund this program were asking us to create a model that was more sustainable and that could help more families. And so with that, we are moving to not necessarily expand how many patients we fund with exome genome sequencing, because as we all know that the costs are declining, but we're going to move in a direction of doing what Global Genes does best, which is evolve to creating a undiagnosed resource hub. And the thought is that we would create kind of a central platform and clearinghouse for families and patients to access next-gen testing resources, what labs exist throughout the U.S. and even globally in a simple way, being able to search along the lines of parameters for turnaround time, pricing, what insurance coverage they take, do they offer funding options, do they offer kind of resources like Rare Genomics where they'll help families crowdfund for to raise funds for their testing, and kind of create a platform for people to tap into this information, and then also support it with somebody who can be available to answer questions, somebody with a genetic counseling background or clinical genetics background. So our status today is to actually put out a request for a proposal. We've got a couple proposals that have come in from individuals who are either a genetic counselor or a kind of master's in genomics. One who we're considering strongly, somebody who has actually worked with Keith Fox and Joe Gleason at UC San Diego on their diagnosed down there, and more to come on this. But just to give you an update, we just want you to be aware that we're moving away from a funding individual patient strategy to a resource hub strategy. And as we evolve this, we'll definitely want this group to be involved in helping us make sure that we're covering all the parameters that we want to cover, and that this becomes a valuable, tool for our community.
Just wanted to jump in real quick and say
Sure.
This is badly needed within the community. I think it's a great way to position global genes, and let me know if you want any help with this. I'd be happy to facilitate in whatever way I can.
Thanks, and we really appreciate that. And we, I think you actually are on the program group, and we've had kind of sporadic calls, but you and Jimmy Lynn and Keith Fox and others, we're definitely going to want your involvement in helping us make sure that we capture all the right parameters and are creating a hub that is that the value that we want it to be. So thank you for your support. So very, very quickly, the last slide on our end was just to kind of share with you, there was a third group, which is called the Partnerships Group. That group was really created to help us evaluate potential opportunities that come up. For those of you that were at our meetings last year, you may recall the Cytosolve presentation where we were trying to figure out, is that a group that we want to partner with? At this point, this group has really only helped us distill down very high level the fact that not only are we trying to create our hub for the undiagnosed world, but we're also trying to figure out a better way to house all our data that allows us to provide what we call a rare concierge service, which is our 1-800 rare helpline. And we have data in lots of different places, and we get questions through the internet, through our website, through phone calls, pretty much on a daily basis, and trying to find the right platform to aggregate that content in a way that's accessible for the rare community. So this is a much kind of bigger project, and we have presented potential platforms such as Apple or even 23andMe, but we're now also looking at a potential collaboration with Genetic Alliance that's in very early stages. So more to come on this. That is where we have looked to that particular group to get input and help us, and going forward to help us vet some of these platforms. So with that, I would like to move to Ethan Perlstein. He is, I love the tagline, leave no mutation behind. He is going to share just kind of the history and what Perlstein Labs is working on, and kind of give you a little sense of his direction, the science, the innovation, and their focus on rare disease. So we're really excited and appreciate Ethan sharing kind of the updates on his work with us today. So with no further ado, Ethan Perlstein.
Great. Thank you so much, Global Genes, for having me here today. So again, my name is Ethan Perlstein. I'm the founder and CEO of Perlstein Lab. We're a one-year-old biotech startup based in San Francisco, and in a nutshell, our mission is scalable drug discovery for rare diseases. If you want to follow up with me at any point after this talk, you can find us online, you can email us, or you can even tweet at us. Slide, please. So I said our mission is rare disease drug discovery, and when I say mission, I mean that in a very specific legal sense. We are a public benefit corporation. So I'm not sure if folks are familiar with this concept, but there's this movement afoot called social entrepreneurship, and one way it's manifesting at the state level is that states are passing these benefit corporation laws, which allow companies to balance, maximize shareholder value with a social and environmental mission. In our case, social mission is the unmet medical need of orphan disease patients. Our environmental mission is to reduce the carbon footprint of drug discovery, and I'll explain a bit about how we do that. But at the end of the day, we are a preclinical drug discovery company, and our beginning point is a mutation. We create models of the disease in simple organisms, and then we conclude at the IND stage and partner with larger developers. And then we repeat, do this for disease after disease. So next slide, please. Again, we're a platform company. We're not a service company. We're not a CRO. Our ambition is to scale across the orphan disease space, in particular single gene diseases, of which there are thousands. We believe we could achieve this scale by using simple model organisms. So that would include yeast, or baker's yeast, as it's sometimes called, which is the simplest of the animal cells. It's a single-celled eukaryote. The next organism of the chain would be the worm, C. elegans, which is about 1,000-cell creature that has primitive neurons and a primitive gut, crawls around with simple behaviors, and sort of represents this early stage in animal development. Then our next most complicated organism would be the fruit fly, Drosophila melanogaster, which has been a model organism for genetics for over 100 years. And then the most complicated organism in our pipeline is the zebrafish, which is a vertebrate. So it has a backbone, it has a liver, it has a brain, but is not a mammal. I joke with people that the only mammals on the team are the scientists. Again, we believe that by using model organisms, we can scale not only across rare diseases, so from disease to disease, but also within each disease, to scale across the mutations that exist within patient populations, because not every person has the same exact mutation and not all mutations are equal. Because we're using model organisms, we're able to get rolling pretty quickly. Again, we started about a year ago, actually April of 2014. And within 6 months, we were able to get all 3 organisms. So we haven't yet gotten zebrafish online, because even though they are much cheaper than working with mice, they're still pretty complicated compared to flies or worms and yeast, which you can get growing on your benchtop pretty much away. So next slide, please. So this is our order of operation from start to finish. So we begin with a mutation. So we select an orphan disease, and we are looking at 2 proof of concept diseases, which I'll talk about. But for this case, we can just say in the general terms. So we can start with an orphan disease, and we can tend to ask, okay, what are the spectrum of mutations that exist? Oftentimes, there can be hundreds of mutations associated with a given disease that are pathogenic. So in our case, we're generally starting with a mutation that results in a complete loss of function, what's called a null mutation. And we often also are trying to look at some of the most common mutations, because even though there are lots of mutations, it turns out that there can be a plurality of patients that have a same common mutation. So in the case, we start with a set of mutations that represent the disease. Then we can take those mutations and program them into these model organisms using CRISPR, which is this new genome editing tool, which allows you to very precisely make edits at the DNA level. And so in this fashion, we can recapitulate a very personalized disease model, because we're taking a specific mutation from a patient and introducing it into these organisms. And how we do that exactly, I'll get into it, but the overview is that the way that this works is that Mendelian disease genes or rare disease genes skew ancient. In other words, these disease genes are present in ancestral form in these simple critters. So you can take a orphan disease gene X, and you can look and ask, is it present in these simple organisms? If it is, then you have an opportunity to use that organism to model. So it is possible that a disease gene is only present in mammals, or only present in higher, more complicated organisms, and it's not present in these simple creatures. Well, in that case, we really can't do much about that. But in the cases where there is this ancestral form of the disease gene in these critters, we can expect that and make these personalized models. And again, so our concluding point, because we're a preclinical company, is we will go all the way in terms of discovery to the IND stage. And at that point, we need to partner with a larger orphan drug developer who can then progress these candidates through trials. And that handoff point at the IND stage allows us, I think, to remain nimble and lean, so that after we've done enough discovery for one disease, we can then go back and do this again for the next disease and rinse, wash, and repeat. Okay, next slide, please. So I don't need to belabor the point about market. Everyone knows statistics, global statistics about rare diseases, I'll just say on this slide that we're focusing initially on this category of diagnosed Mendelian diseases. So what would an example of that would be, cystic fibrosis. The gene CFTR that was responsible for cystic fibrosis since 1999, and there's about 1,700 other Mendelian or single gene diseases where we know what the causal mutation is, and we often even have a database of mutations that have been observed in patients. But there are an equal number of diseases that appear to be Mendelian. In other words, they appear to be caused by a single gene based on looking at families and looking at inheritance patterns, but we still haven't figured out what the genetic culprit is. Every couple of weeks you hear, or maybe even every week, you hear a case of an undiagnosed disease moving from that category to the diagnosed category, and that's through the help of genome sequencing and the reduction of cost there, which enable these undiagnosed diseases to finally have a genetic culprit behind them. So our initial focus will be on Mendelian or monogenic diseases where we already know what the genetic cause is. But eventually we think that any single gene disease where that disease gene has an ancient form in these simple critters, we think that makes them eligible for study and platform. Next slide, please. So what I mean by a precision orphan drug, we are really thinking about small molecules. We're not doing antisense. We're not doing gene therapy. We're kind of doing old school pharmacology, you could say. And when we look to an example of what we mean by a precision orphan drug, there's no better case than Kalydeco, which is a cystic fibrosis modulator, which is a wildly successful drug, both in the clinic and also in terms of actual sales as well. And there's all separate question about orphan drug pricing, which we won't get into, but I only bring this up to show you what kind of scale is in terms of not only what the value of such a drug is, but also what size of population of patients we're talking about. Because cystic fibrosis is a complicated disease in the sense that there's over 1,000 mutations in the CFTR gene. And so Kalydeco is not a wonder drug for all cystic fibrosis patients. It's only been approved for specific mutations, but that's okay because that's exactly the kinds of precision molecules we want to make that work on a mutation-specific basis. Next slide, please. And so, as I mentioned earlier, why model organisms and why rare disease? Well, I think that the answer is that, as I alluded to briefly, if you look at the genes that are responsible for orphan genetic diseases, they skew ancient. And here's an example of what I mean by that. So we take here a prototypical cell. This would be a eukaryotic cell, of course. So this would satisfy. So yeast would meet this condition, and even obviously human beings would meet this condition, even though there's a large number of differences in the cells of the total organism. At actual cellular level, the plumbing and the guts of the cells, whether it's a yeast cell or a human cell, are actually quite conserved, especially when you look at the lens of the architecture of the cell. If you look at these various compartments that you might have learned in high school biology, the ER, the Golgi, the mitochondria, the peroxisome, each of these conserved structural features of eukaryotic cells has orphan disease associated with them. So this is really what to us solidifies this good fit between model organisms and rare diseases, because we think rare diseases fundamentally are many of them involve ancient cellular biology. Okay, next slide, please. Let's look at a particular group of diseases to really illustrate this point about evolutionary conservation. So what this is showing you here are, you can look at the genes responsible for each of the 49 lysosomal storage diseases and ask, how are these disease genes or homologs conserved across the simple organisms, yeast, worm, fly, and zebrafish? So if you start all the way on the bar graph on the right, where you see Niemann-Pick, bat disease, and Walman disease, so what's in common between those three is that the ancestral form of the Niemann-Pick C1 gene, which is responsible for Niemann-Pick C in people, that gene has an ancestral form or an ortholog in 4 out of 4 of the organisms. So that's what we would consider a really ultra-conserved disease. And if you kind of march to the left in this graph, you can see that the distribution here is telling you that if you go all the way to the left, this is the list of genes, which is the lysosomal storage disease genes, which you can only model in the zebrafish because there is no ancient form going back any farther into simpler critters. And then you get sort of combinations in between where you can model a given disease in, say, worm and fly, but not in fish or all these other different combinations. But in general, this pattern of finding that there is conservation of these disease genes across organisms is conserved, even if you might find in each individual case, you might find that this organism is slightly better matched than the other organism. Slide, please. Our first one, mentioned before that we are building a platform company, but we need to first prove the platform in a couple of cases. And so our first proof of concept disease is called Niemann-Pick type C. Some folks are familiar with the disease. It's gotten a lot of press as rare diseases go. It's considered an ultra rare disease. And for a lot of other scientific reasons, it was a good fit for us. And one of the principal reasons why it was a good fit for us as a proof of concept is that academic groups, going back even as far as the year 2000, had found that there are ancestral forms of the Niemann-Pick C1 gene in these simple critters. And some of these academic groups went so far as even to knock out the ancient form of the gene in these organisms and show that these critters get disease-like phenotypes. In particular, the inability to develop normally, and in other words, these critters die early in development, and that's actually something that we exploit in our drug screens. Okay, next slide, please. A little bit of detail about Niemann-Pick C. So one of the major characteristics of the disease is neurological decline, and if you look at a cellular level, what seems to be going wrong, and then there is actually still some contention about what are the exact set of effects that take place to cause disease, but one of the hallmarks at the cellular level is an aberrant, in other words, a pathological accumulation of cholesterol and other lipids. And so that's actually diagnostic of the disease. If you take a sample of skin cells and you want to diagnose, you find the characteristic of lysosomal storage, which is actually a cellular feature that's shared across these diseases. But the exact specifics of what's going on in the disease are not terribly important. For our purposes, what you need to know is that when these simple organisms get Niemann-Pick C, or when we give them Niemann-Pick C by introducing mutations in their gene, they manifest these developmental delays, which is actually something that you see in patients. Next slide, please. Is our flow, and this is a template that we've used for our first disease, and this template we'll use as we plug in additional diseases going forward, but hopefully we'll be accelerating the times it takes to move through each of these steps. So the step in any disease program that goes through our platform is to develop the models. And as I said, you have to satisfy a few conditions to be eligible for our platform. The chief among them is if you do a sequence comparison and you look at the genomes of yeast, worm, and fly, and fish and compare them to human, can you find an example of the ancestral form of the gene? And if you do, the second experiment you want to do is you want to know that if you were to knock out the gene in the lower organism and replace it with the human form, you would actually get back normal function. In other words, you actually have to prove that not only is there this sequence homolog, or in other words, just by DNA comparison, you can see that there is an ancestral form. That's not enough. What you actually have to do is do a function test. Can you actually swap the two forms of the gene, the ancestral for the human, and show that you actually get interchangeability? So I think we did in the very beginning to make sure, and again, with Niemann-Pick C, a lot of that work of validating that the Niemann-Pick C gene is functionally conserved was done by academic groups, so we were able to hit the ground running. So the first step, and the next step, we developed assays using sort of standard high-throughput screening technologies, and this means using multi-well plates, so 34-well plates, 96-well plates. It means using liquid handlers and other robots that allow us to do rapid screening. All of our assays involve whole animals. So put the whole animal into the well of these multi-well plates, and then we're able to read out the rescue of developmental delay. Because in this case, what we're looking for is, can we find a small molecule that prevents these Niemann-Pick C worms and flies and yeast from dying prematurely? And so our platform, so maybe just click through because there's just animation here. So here, this is actually looking at what I mean by our high-throughput screening platform. So it's really based around 4 instruments. We have the ability to do liquid handling manipulation, so we can move very small volumes around and very large volumes around into these plates. We have a machine called a biosorter, which allows us to take individual worms, individual flies, and spit them to the wells of these plates so that we can manipulate organisms. We can manipulate these animals on an animal-by-animal basis. And then we have a microscope and a box machine that allows us to do very rapid and automated visualization and data analysis. We began our Niemann-Pick C screening campaign in January. We have a 50,000 compound pilot library collection that is enriched for drug-like and lead-like molecules, and we're about 75% done with the screen. We anticipate finishing the screening campaign in the end of next month. And once we've completed that screen, we will take these hits from these lower organisms and then validate them in a number of assays on the road back to patients and higher animal models, more conventional pharmacological and disease models. So we have patient cells that we're growing in the lab, both immortalized cells and also stem cells. There are a number of NPC mouse models available, and so all of the hits that we find in these low organisms will be translated to the either patient cellular models or mammalian disease models. And that work will begin in the second half of this year. Advance, please.
Before we,
sure.
Ethan, just want to draw attention that it is the top of the hour, and we know that we ran long, so we would just like to ask if you are able to stay on the call for another 10 minutes.
Sure.
And
yeah.
allow Ethan to finish his presentation, and then we'll just give one housekeeping reminder and let everyone go on their way. So if you're able to stay on the call, we'd really appreciate it.
I'll wrap it up super quick in a couple minutes. Okay, next slide, please. So let me just talk a little bit about the actual models that we use for yeast. We have a model based on growth reduction and then rescue by a small molecule. And in this case, we recreate Niemann-Pick disease pharmacologically as opposed to genetically, but don't really need to get into the specifics of that. The thing that you need to take away from this is that we have yeast cells that are not growing, and then we look for a small molecule that allows them to grow. So those red wells there represent cases where the cells are no longer dead, they're now viable and they're growing, and presumably there's a molecule in there that has rescued them. So that's the yeast model. We can move on to the next slide. We have a C. elegans or worm model. So when we knocked out the NPC1 gene in the C. elegans, what you can see is that there are fewer worms and they develop more slowly. So if you're comparing the image on the left, which is a Niemann-Pick mutant, you can count the worms, and then you can see on the right, which is a normal population, they're full of worms. And so in this screen, we're looking for a small molecule that can make the well on the left look more like the well on the right. And the two panels on the bottom are just our overlays that we do analytically so that we can count the number of worms, measure them very accurately, and see that we're getting a developmental rescue by a small molecule. That's the C. elegans model. Next slide, please. We have a model. So what you're seeing here is a time course of fly development. So flies start as embryos, just like any animal, then they become larvae. These larvae are kind of like human infants, and I know what that means because I've got one at home now. And so what these little larvae do is they develop to a certain point, and then they metamorphose into adults. And what happens in the case of Niemann-Pick mutant flies is that they don't develop normally. They arrest after about 24 hours, and then they stay small, and then they die. And so in this screen, we're looking for small molecules that can make these developmentally arrested larvae normal again, make them either alive or develop further. Okay, next slide. We're doing, obviously, by screening in parallel, is looking for intersections of activity. So we've done a preliminary analysis on our screen so far, and we find that our hit rate is low overall, which is good. We have a specific assay, but we're also finding that there are instances of overlap. So we're finding cases of a molecule that can rescue the fly NPC model and the yeast model, but not the worm model, and vice versa. We can find molecules that are rescuing the worm and the yeast models, but not the fly model. And we actually do find cases, rare cases where that's not reflected in the slide, where we find rescue across all three, but that tends to be the rarest category. Next slide. And here's a glimpse of our functional validation. So we have a patient cell line that is, we can stain with a dye for cholesterol. I said before that one of the hallmarks at the cellular level of Niemann-Pick is this accumulation of cholesterol. And so all those hits from those previous screens, we put onto here as a sort of first sanity check to make sure that these molecules are actually working in the context of a human cell, and that they're correcting a phenotype that we know is very specifically associated with the disease, if not diagnostic of the disease. Next slide. This is the first version of the platform. The three organisms feed into this conveyor belt here of instruments that allows us to do these rapid screens in parallel. We do that validation from the cell models, and now we're working with a potential mouse partner so that we can translate directly from hits that are active in these lower orgs plus cells right into mouse models. So this first version of the platform. Next slide. We want to do what we'd like to do to really achieve the platform is to parallelize this. And so because this is really just based on a number of instrument clusters and expertise on these organisms, we think that we can very easily parallelize this and build up this platform to now address multiple diseases concurrently, and of course multiple mutations of each disease concurrently. Next slide. So just go through this really quickly. We're going to complete the Niemann-Pick screen by June. We want to file provisional patents on the small molecules that emerge from the screen. We're not interested interested in repurposing. We're interested in novel chemical entities. We are ultimately looking to find preclinical drug candidates that we want to validate in higher animal models, which we think is going to be a sort of contingency for partnering with larger developers. And we're already in discussions with drug developers that have expertise and drugs already in their portfolio for lysosomal storage disease, which we think would be obviously ideal partners for this particular program. And then the last slide, I think. Actually, we can. The next disease is on deck is called NGLY1, and I can get to that offline. But the point is, we believe that the platform we built for Niemann-Pick C, we can plug and play and very quickly make this work for other diseases. Next slide. And then just a brief glimpse of our team. We're all former academics. We're all first time in a company, but the team was really built around expertise in these different organisms. So Nina is an expert in patient cells that actually done work in gene therapy in grad school. Tom is an expert in flies. He's our resident Drosophilist, and Sangeeta is our resident worm expert. And we have advisors who have got decades of experience either in medchem or business development helping us along. And we have a stellar board, including Matt Wilsey, whose daughter has NGLY1, and he's been a tremendous support. I think that's it for the presentation. And if you want to read up any more about us, we've gotten some nice press. And if anyone wants to follow up specifically on Niemann-Pick C, please do so offline. Thank you.
Thanks so much, Ethan. That was a wonderful presentation and was really enlightening. And I hope that others on this call will follow up with you. I don't think we have time for questions today, but you all should have Ethan's email address and have access just to his website, so you should be able to follow up with any questions after the call directly with him. And I apologize for not having time for questions today. So I'm just going to up our call today. We obviously try to a lot of information into this hour. We will be following up with you with Nicole committed the current patient summit agenda to get input from you. We'll also provide the rest of the group the mobile screener app kind of concept statement, so you can see where that is. And then just some housekeeping items. Please note that we've scheduled the call. We haven't sent out an invite yet, but we're aiming to have our next quarterly call June 24th at 10 a.m. Pacific time, 1 p.m. Eastern time, and then look forward to seeing you for the next in-person meeting, which will be again Saturday of our summit tribute weekend at lunchtime, as it was last year. So more to come on that. Thank you all for your time and for indulging us for an extra 5 minutes. And Ian, thank you again for your wonderful presentation. If we do not have any last comments or questions, we will adjourn our call. So final comments or thoughts?
Thanks, Andrea.
Yes, thank you.
Thank you all. Have a good day.
Bye-bye. Thank you.
Thanks. We're adjourned. Bye-bye.